Welcome to this second discussion on the nervous system. In the previous conversation, we looked at an introduction to nervous tissue. We looked at the neuron, learned about the parts of the neuron, took a look at glial cells, the 6 different types of glial cells. Also took a look at a couple of different ways that the nervous system is arranged. Think structurally the PNS or the CNS. And then we looked at functional differences in sensory versus motor function. In today's discussion, we're going to be looking at Blossom 13.1 and 13.2. We'll spend just a moment looking at the embryological development of the nervous system. It's always important to be somewhat mindful of how these systems and tissues are formed. And then we'll spend most of our time looking at 13.2 on the central nervous system, where I will introduce to you the brain and the spinal cord. Now, the brain and the spinal cord and the neurons from last time are significant components of your Lab 6. So when you get to Lab 6 after the lab exam, you will then be dealing with the nervous system. That's our one and only lab on the nervous system. And that lab is going to cover a number of things which will look familiar to what you're learning in this discussion here today. So let's take a look first at 13.1. I have gone ahead and I've already highlighted a couple of key terms, and I have blocked out some things that are not as important. But let's take a look at what the nervous system, or where the nervous system comes from. And we already saw this early in the course. We learned that the nervous system comes from the early embryo in the outer layer of the ectoderm. So in the early stages of development, the early embryo, the gastrula, is made up of 3 layers. The inside layer, the endoderm, is the layer from which all the tubes, like the digestive system and the respiratory system, are derived. The mesoderm, remember, is that layer which is going to form all of your muscle tissues and connective tissues. And then the ectoderm was the outer layer, and it goes on to form your epidermis, and that makes sense, right? The outer layer of your body, outer layer of the embryo, but also the ectoderm, I think perhaps not intuitively, also forms the nervous system. So let's take a look, not at some gritty details of this, but let's just get an idea of how this happens. So again, when conception occurs and egg and sperm come together, we get that fertilized egg. That fertilized egg, recall, is called the zygote. That's a term that you should certainly recognize. And the zygote is that one cell, right, from which all of our cells are derived. We've discussed this when we talked about early development. We talked about mitosis, right? This cell undergoes mitosis to make 2 cells. It undergoes mitosis again to make 4 cells, 8 cells, 16 cells. Eventually, that ball of cells called the morula begins to form 3 germ layers. And those are the 3 layers I was just describing. And we discussed the word, or described the word, differentiation. So as these initial layers are formed, these cells begin to undergo layers of differentiation where they become different from each other to eventually form all of the different types of tissues and cells in our body. So again, like I just said, here it is in writing, the endoderm, right, goes on to form those tubey-like things like your digestive and respiratory systems. The mesoderm, the middle layer, goes on to form your connective tissues and your muscle tissues. And then the ectoderm, that's where we're focusing right now, goes on to form the integumentary system and the nervous system. So just keep that in mind, that the nervous system is derived from the ectoderm. So again, the blue areas we're going to not worry about. It kind of describes the very, very earliest signs of how this occurs, and we're not going to go into that embryology. But just know that at about the 25th day post-conception, the nervous system is now broken up into 2 basic parts. You have the anterior portion and the posterior portion. The anterior portion will eventually become the brain, and the posterior portion of this early nervous system will go on to form the spinal cord. Not hard to imagine as you think of the brain being more anterior and the spinal cord being more posterior. So that anterior part, the part that's going to become the brain, undergoes some swelling, some enlargement, some sac-like structures. And these sac-like structures are called vesicles. And they all have the same root name. They're all called encephalon. Encephalon. And you should recognize that en means inside, like endo or en, and that cephalon or cephalon means head, right? A cephalic symbol, or the cephalic region toward the head. So let's take a look at these words. Prosencephalon. Pros means in front of. Mesencephalon. You know that mes means middle. And finally, there's the rhombencephalon. You might recognize this term from geometry class, a rhomboid, right, or a rhombus. And that is a four-sided figure, if you will. We can also divide the brain up, though, into three different areas. And we'll see this as we go through these images. There is the forebrain. Okay, there's the forebrain. There is the midbrain. And there is the hindbrain. So we'll be looking for those ways as well of dividing up the brain: forebrain, midbrain, and hindbrain. Okay, so let's take a look at some images of those. Now, once those primary vesicles, those first areas of swelling, are formed, there are a couple more that form, and it turns out that those three primary vesicles become 5, quote, secondary vesicles. And here we're going to see a couple of terms that you're going to see over and over. We're going to see the telencephalon. Now, you know from our vocab that tel, think telophase, right? So telophase, or the telencephalon, this will become the cerebrum. You know, when you think of the brain, you are probably thinking of the cerebrum. The cerebrum is the big fleshy part of the brain. It's the part that contains the lobes, named after the bones that protect it. So the frontal lobe, parietal lobe, temporal lobe, occipital lobe, all of those lobey, squishy parts of the brain are the cerebrum, and that is referred to as the telencephalon. It is developmentally one of the last parts of the brain to form. So it makes sense. Telencephalon, last, right? The last part. So it's one of the last parts that develops, and we'll discuss later in this course, maybe even in this chapter, that there are parts of your frontal lobe that are not completely formed until you're age 25. So clearly our brain is still developing. The telencephalon, or parts of it, are the last regions to develop. The other name here, the other term here that you're going to hear a lot about is the diencephalon. Now this name, diencephalon, it's referring to basically the center, the core of the brain. When I think of the brain, and you'll hear me describe this, I think of a peach, right? A peach. So you have the edible, fleshy part of the peach. The part of the peach that you can eat, that is the telencephalon. Those are the lobes. But we all know that in the center of a peach, there's a pit, a core, something that is not edible. That, if we're talking about the analogy, that's the diencephalon. So the diencephalon is the pit, the core, the center of the brain itself. And so we'll see that word diencephalon, and in the diencephalon is where you have a couple of important structures called the thalamus and the hypothalamus. And we'll learn about those and learn to label those as we go through this. The mesencephalon doesn't change names. It is still the midbrain. This is a relatively small part of the brain, sort of connects the front with the back. And then finally, that rhombencephalon that we saw develops into two other areas, the metencephalon and the myelencephalon. Now, I know these look really, really similar to each other, and they are. The metencephalon goes on to become the pons and the cerebellum. Again, we'll label these and learn about the pons in the cerebellum. And finally, the myelencephalon goes on to become the medulla oblongata. Okay, the medulla oblongata. So this is not perhaps making much sense to you yet, but it will. As we go forward, I would recommend that you just kind of listen back, look at these areas again once you better understand these structures within the central nervous system. If we're talking about the forebrain, hindbrain, midbrain regions, this, right, the Telecephalon and the Diencephalon, this is the forebrain. The midbrain, like I said, is a very small area, and the hindbrain would be part of what we're talking about here. It would be the brainstem, which includes the pons and the medulla. Here's a visualization of this. I'm not going to ask you to label this, but here is a representation of the early embryo. Okay, the early embryo, and the part where we see these three swellings, right? The prosencephalon, what becomes the forebrain, the mesencephalon, the midbrain, and the rhombencephalon, the hindbrain. And then as we go a little further in development, we see that those three areas split into 5 areas. Okay, again, I'm not going to ask you to label this, but those 5 areas are here. What I do want you to know, however, is what is the telencephalon? What does it become, right? The cerebrum. What does the diencephalon become? It becomes the thalamus and the hypothalamus. The mesencephalon is the midbrain. The metencephalon is the pons in the cerebellum, and the myelencephalon becomes the medulla oblongata. These are the adult structures that you will be learning to label in the brain. So by the time you are born, your brain clearly forms or has those 3 main areas. You can clearly differentiate a forebrain, a midbrain, and a hindbrain. But there's still a lot going on after birth. So here is a lateral section through the brain, sort of a mid-sagittal slice. And let's just take a look. The pink region, again, the area that you think of as being the brain, you could call the forebrain. The midbrain is a very small connector piece, and then the hindbrain is going to be the cerebellum and the pons and medulla oblongata. If we want to take a look at this in a different way, those are the 3 regions. If we just look at where I'm pointing now, the fleshy part of the brain, that's the telencephalon. The pit, the core, the center of the brain that I'm circling now, this would be the diencephalon. The diencephalon. Right here in the center, this region that I'm circling now is in fact the thalamus, and this region here that I'm pointing to is the hypothalamus. Again, we're going to be diving into those regions more carefully in a few minutes. It's a nice little video here about how this all goes together. I think that'll be interesting for you. And then finally, the spinal cord. Remember that the anterior portion of the neurotube is forming the brain. The posterior portion of that early tube is forming the spinal cord. And it's not as fancy. It doesn't have all these different areas as much, but just know that it's more on the dorsal side, right, of the body, of course. As you take a look at the arrangement of the brain and the spinal cord, you can see that there is quite a flexion, a turn here in the directionality of information, and that's important. If we had sort of the same straight arrangement of 4-legged animals, we'd be looking straight up, wouldn't we? So we have this, what's called this, the cephalic flexure, which is basically a turn in the overall nervous system. As we come up the spinal cord, we are then looking forward, of course. And that, my friends, pretty much brings us to the end of this little introduction to the embryological development of the nervous system. This table, this is table 13.1, does put into a table the different stages. So again, the primary vesicle stages, the secondary stages, and what we find in the adult. So again, the prosencephalon branching to become the telencephalon and the diencephalon. The telencephalon is the cerebrum. The diencephalon is still that area, better known as the thalamus and the hypothalamus. And you can see the table here. Again, gives you just a different way of organizing the material that was described above. So that brings us to the end. Here's a really interesting story. I'm not going to quiz you on this, but you may know of someone who has been born with a neurotube defect. Spina bifida is probably one of the most commonly occurring one of these disorders. And basically what happens is that in development, the spinal cord is not completely enclosed, and there becomes an opening to the outside world. And on the left here, this is normal, right? So here's the spinal cord completely protected inside the vertebral canal. And there are situations where there's a break in this or a complete opening. And this can lead to paralysis in these areas. So depending upon the severity of the opening, will tell you whether or not the complications that will be present. This is a condition which is present on an ultrasound. So one of the many things that that you're looking for in those ultrasounds taken during pregnancy is you're looking for a bubble or a bulging here in the lower spinal cord. And they are even doing some amazing things now. They're actually able to go in during labor, not during labor, during pregnancy, and they're able to go in and sometimes correct these bubbles or correct these defects, and the baby is then born with far fewer complications as a result. So that's the end of 13.1, and the most of our time is going to be spent here in 13.2, where we're looking at the brain, looking at the regions of the brain, looking also at the internal regions of the brain, and then finally taking a look at the spinal cord. We'll also be looking at the arrangement of gray matter and white matter. Recall I discussed that last time. White matter in the central nervous system is tissue that is rich in myelinated axons, and then the gray matter, recall, are areas that are rich instead in cell bodies and dendrites. So let's take a look. Again, keep saying it, but make sure you're watching these videos, taking note of what you're learning. And let's take a look at the brain. Again, this is the cerebrum, the majority of it. What's highlighted down here is the cerebellum. Watch this video and it will help you remember that. I remember the cerebellum as the little bell, right? The little bells that are hanging from the posterior portion of the brain. So let's start here with some basic anatomy of the cerebrum. The cerebrum, like other solid organs, think a brain, brain sort of like being like a bone. Remember that the outer layer of a bone is also called the cortex or the cortical region, and the inner region of a bone, where the bone marrow was or is called the medulla, right, or the medullary region. So same is true in the brain. The outer region is the cortex, and remember that the cortex is also, we saw this in the actual image of the brain, the cortex is the gray matter, and we'll see that again as we go through this. The cerebrum is divided into two large hemispheres. So there's a left hemisphere and there is a right hemisphere. So a left hemisphere and right hemisphere, each half. Each half is separated by a deep groove called the longitudinal fissure. Okay, now you're learning in the bones, and you're going to continue to see there are two common names for grooves. A shallow groove is oftentimes called a sulcus. A sulcus is a shallow groove, S and S, whereas a fissure is a much deeper groove. So this longitudinal fissure is very deep, and it's named appropriately, and it's running along the mid-sagittal plane. That is the longitudinal fissure. If we go deep into the brain, we will see that there's an area of white matter that connects the left and right hemispheres together. It is outlined here, but this would only be visible if you had a mid-sagittal cut, and this big structure that connects the halves of the brain is called the corpus callosum. The corpus callosum. I always remember the colossal structure. Remember, corpus just means body or structure. Colossal, callosum. So this is a big old structure or body that connects the two halves of the brain, and you'll see it in other images, but it is white. It is white in color. So what does that tell you if it's white? That's right. If it's white, it's telling you that it's axons, right? So this is basically a big band of axons that are connecting the two halves of the brain. We're also going to look inside the brain, and we're going to take a look at some regions of the brain. Inside the brain is an area referred to as the limbic system. It's not a body system like the respiratory system or the cardiovascular system, but we still put that word on it, the limbic system. It's a series of structures that we'll learn very briefly that have to do with emotion and memory and behavior. If you've taken a psychology course, you likely discussed the limbic system as part of your emotional, memory, behavioral centers of the brain. That is deep down inside, okay, inside. So let's take a look a little bit more at the cortex. Again, the cortex, if you look at it, it looks like a big wrinkled, it's on the outside, it's wrinkled, right? Each of those wrinkles, or each of those noodles, I call them noodles, is called a gyrus, many gyri, right? Those are the big wrinkles or the big noodles. And then separating those individual noodles are shallow grooves. Each one is a sulcus, and we would say that there are many sulci. So the surface of the brain, the cortex, what you visibly can see is a series of gyri and sulci. Take a look at this picture, and they're not labeled here, but you can see, in fact, that there are these shallow grooves. Each one is a sulcus, and these big noodle-like structures, these big wrinkles, are the gyri. Now, each of these gyri has a name. Thankfully, we will only be naming a couple of them in a few minutes. Most of them, we're not going to name them, although, trust me, there are names for them. What I want you to see in this picture is that there are lobes of the brain. Okay, now keep in mind that this is only one half of the brain, right? We're just looking at, well, you tell me, which hemisphere are we looking at here? That's right, this is the left hemisphere. Here's the cerebellum. Here's part of the spinal cord coming down. And the cerebellum is always toward the posterior or dorsal side. That's one way of remembering this. The other way that I look at this is that there is a very deep groove right here that is found more anteriorly. So this is the anterior side. So we're definitely looking at the left hemisphere. And what we can visibly see in the left hemisphere are 4 lobes. There's the frontal lobe, and you already know that this is largely protected by the frontal bone. There's the parietal lobe, again protected by the parietal bones. The temporal bone protects the temporal lobe, and the occipital bone is protecting the occipital lobes. Now remember, there's only one occipital bone, but there are 2 lobes, left and right, and there's only one frontal bone, but there are 2 frontal lobes, left and right. We see here just a little bit of extra information popping out of here. This is the olfactory bulb. We'll see this later on. This is one of the cranial nerves. This is cranial nerve number 1, and this is the nerve. It pops out of the brain. It's responsible for your sense of smell or olfaction. And imagine that your nose is right about here, right? So your nose is sitting right here, and the smells that you bring in through your nose are picked up by this nerve and travel to the brain. a couple of additional structures. And again, all of these are really important structures that you'll be seeing in Lab 6. The lateral sulcus, right? Lateral, on the side, sulcus, a groove. This is this one. So as I point right here with the pointer, this is the lateral sulcus. The lateral sulcus is a very distinctive groove that separates the temporal lobe from the other regions, okay, from both the parietal lobe and the frontal lobe. I'm just going to take advantage of this picture, this still frame of this video. This in blue is the frontal lobe, but I want you to see right here, this groove right here. This is part of that longitudinal fissure, that very, very deep groove that separates the left from right sides and also separates the left frontal lobe from the right frontal lobe. On the side here, we're seeing the lateral sulcus. You can see that here. So that makes what I'm pointing to now the temporal lobe. And back here, this would be part of the parietal lobe. The occipital lobe is not visible in this particular view. Let's take another look. Okay, now we're still looking at the left side of the brain. I'm going to add just a couple more features to what we just learned. So we see the frontal lobe, parietal lobe, temporal lobe, occipital lobe, that's the same. We see the cerebellum, and this particular image has two other brainstem structures labeled, the pons and the medulla oblongata. Go back when you get a chance and look at which embryological layer forms both the pons and the cerebellum, and you'll see another one of those secondary vesicles forms the medulla oblongata. And then where my pointer is down here at the very bottom, the medulla oblongata is continuous with the spinal cord. Here is that lateral sulcus as we get our bearings. And there is another sulcus referred to as the central sulcus. The central sulcus is significant in that it divides. It is that divider between the frontal lobe and the parietal lobe. Central sulcus. So let's go back up to the previous image and find the central sulcus, and it would be this one right here. Okay, so it's not labeled, but this would be the central sulcus. You can clearly see, and it's really easy to appreciate it here because the artist has made the different lobes different colors. But you can see that this is the divider between the frontal and the parietal lobe. That's the central sulcus. Now notice that there are two very distinctive gyri right on either side of that central sulcus. Those are the two sulcuses, or the sulci, that you are responsible for. The one that is just in front of the central sulcus as a part of the frontal lobe, this is the precentral gyrus. Precentral gyrus. And then the gyrus, which is just behind the central sulcus as a part of the parietal lobe, this strip of land is called the post-central gyrus. We'll see the significance of these as we move forward, but these will be important landmarks for you to learn how to label precentral gyrus, post-central gyrus. Here's a little rotatable 3D model that will help you appreciate these structures in 3 dimensions. You can spin and rotate this model as well. Now, many years ago, there was a German scientist by the last name of Brodmann, and he extensively studied the brain. We know so much more now about the brain than Dr. Brodmann did back in the early 1900s. But he started classifying parts of the brain based upon all sorts of data that he was collecting, and some of these areas are still described using his nomenclature from over 100 years ago. So I'm not going to have you memorize the area number, okay? I'm not going to have you memorize the area number, but I do want you to see that these areas many of them do correspond with a number of these regions that you'll be naming. So for example, not to memorize it, but area 4 we now would call the primary motor cortex, and that primary motor cortex is that precentral gyrus. So this green strip here is that precentral gyrus, and then the postcentral gyrus, right, this sort of turquoise strip of land, right, was another area that he described using different numbers. So I'm not going to go into these names yet, but we will be naming some of these things. For example, we will learn about Broca's area here, or learn about the primary auditory cortex. We will learn about these areas, but we will not be calling them by their number that he described. Although neuroscientists and neuro physicians still do use these areas in their conversation. So let's take a look at a couple of these areas. Let's go post-central gyrus first. The post-central gyrus, okay, this strip of land in the brain is also called the primary somatosensory cortex. So let's make sense of this. Cortex just simply means an area on the outer region of the brain, right? It's one of the gyri, one of the bumps. Primary, the major, the most important, and sensory is what I want you to see here. So the post-central gyrus is also the primary sensory cortex. You'll also see this region called, this over here, sorry. See it called the sensory strip. If I can get this to work, sorry. So the post-central gyrus, if I can just spell, is also called the sensory strip. Okay, so it's a strip of the brain, right? The strip of the cortex, and it's where most of your sensory information is perceived. So we'll leave that there, and we'll save that little comment for your notes. This is the area where you're feeling, right? You're feeling all these things that come in from your body. So senses, right? The fact that you can feel pressure and touch and pain and vibration, all of those regions go up the spinal cord and go right to this region, the postcentral gyrus, and we'll see this as we go forward. The precentral gyrus is the primary motor cortex. This region is also called, so the precentral gyrus is also called the motor strip. Okay. Now the motor strip is, remember, on the frontal lobe. And so this is the region of the brain that is going to be sending down from your brain planned movements, voluntary movements. So when we see the precentral gyrus, again, it's a part of the frontal lobe. This is the part of the brain that sends signals that allow you to move your skeletal muscles. Your voluntary muscles are controlled by this region of the brain. We'll look at that as well. A couple other areas that I want you to know well. One is Broca's area. This is the area that is responsible for controlling the production of language, or the movement of structures responsible for speech. Think, let me get a different color. Think your tongue. Think the coordination of your voice box and all that's going on in your throat. Your ability to speak is one of the most complex muscle movement neurological phenomena that we have in our body. And so this area, Broca's area, I'll show it to you in a moment, is an area that is controlling this production of language, or the movements required for speech. If a person has damage to this area, they would not be able to produce speech in a quick way. Their speech would be negatively affected. The other area also related to speech is Wernicke's area, and it is related to the perception of speech. So A person who has brain damage to Wernicke's area would just not be able to follow along. They can hear just fine, right? Their hearing is fine, but they're not able to process and understand what is being said to them. So you may know people who've had strokes, and if, for example, they're just not understanding what's being said to them, it's very likely that their Wernicke's area was affected by their stroke, and they're just not able to process what they're hearing. Whereas if a person who's had a stroke understands everything fine, but when they go to speak, it's garbled and just doesn't make any sense, and they have difficulty getting the words out, likely their stroke affected their Broca's area. These two regions, Broca's area and Wernicke's area, are found on the left side. Okay, so what we're also going to learn in this unit is that the right side of the brain and the left side of the brain are not simply mirror images of each other. We're going to be seeing this a lot. I think that there's sort of a perception that externally, right? Let's face it, you look in the mirror, we look very symmetrical. We have 2 arms and 2 legs, 2 eyes, 2 ears. Our body is largely symmetrical from the outside, right? There's a left and a right of everything. However, when you get inside the body, you start looking at organs, right? We don't have 2 livers, 1 on each side. We have 1 liver, we have 1 gallbladder, we have 1 pancreas, 1 stomach. We begin to see that there is not this parallel universe inside of our body. Even more stark is that the brain is not two halves of the same thing. There are some structures, some responsibilities only found on the left side of the brain. And here's an example. The left side of the brain is, for most individuals, their speech side of their brain. And the right side of the brain has other responsibilities. So they're not mirror images. We'll see the same thing when we get to the heart. The heart is oftentimes described as being a double pump. The left and the right side of the hearts are vastly different. When we see the lungs later on, the left and right lung are not the same size. They are actually significantly different in size. Your 2 lungs are not the same. Your 2 kidneys are not exactly the same side by side. Even though you have 2 kidneys and they're separated from each other, they're not the same. They're anatomically shifted one from another. So we will see this idea of asymmetry as we look deeper into the body. So in this video, 13.4, it is going to describe to you Wernicke's area and Broca's area. Definitely know these areas. I can tell you now that they are important regions of the brain, and I will ask you to know these areas well on the exam number 3. So let's take a look at another brain. Again, we're still looking at the left hemisphere. This particular drawing doesn't have the cerebellum. So you don't have the cerebellum down here where I'm pointing as a reference point, but you do have that lateral sulcus, which is coming in from the front, and that is the greatest landmark in this view. The other landmark, now that I'm going to tell you about it, that you know that this is the left hemisphere, besides just its orientation, is here. The artist has circled Wernicke's area and Broca's area, and these are in most individuals on the left side. I say most people. There are individuals for whom the Broca's area and the Wernicke's area are on the other side of the brain. And there are people in this world whose heart is essentially backwards and whose organs are reversed, left versus right. Functionally, everything is good. They have both sides of the brain, they have both sides of the heart, all is good, but they are different from the majority of people. So there is some anatomical variation here. But we'll use the normal scenario in describing the Wernicke's and the Broca's areas as being on the left side. So again, we see the lobes, right? The pinkish area is frontal, the bluish area is parietal, green for occipital, and the tan is the temporal lobe. But there are some extra features here. Let's go back. Here is that precentral gyrus. We're still in the frontal lobe, and it's also referred to as the primary motor cortex, and like I said, it's also called the motor strip. And then This line, right, this would be the central sulcus dividing the frontal and the parietal lobes. And then here, this strip of land, this is the post-central gyrus, aka the primary sensory cortex, also called the sensory strip. The parietal lobe is a lot to do with sensory. So in addition to the sensory strip, the rest of the parietal lobe is referred to as the association area. An association area is on the brain an area that contributes to your senses, right? In this case, this is the area of the brain that contributes to your sensory components, but your primary sensory area is here on the postcentral gyrus. Same thing in the frontal lobe. You have the primary motor cortex, but there's another part of the brain that's also contributing to motor control, and it's referred to as the association area. You might hear about the prefrontal cortex. This is a region of the brain, very interesting. This is the last part of the brain to form. I mentioned it doesn't form until age 25. This is the part of the brain that is responsible for executive functions, for thinking. When a person, when a young person is asked, why did you do that stupid thing? And they say, I don't know, right? Oftentimes, honestly, their prefrontal cortex is not completely formed yet, and they really aren't able to fully appreciate or grasp some of the bigger consequences, some of the consequences of their actions. And again, this is about age 25. Now, where is this Broca's area? Broca's area is right here. It's in the frontal lobe. It's in the frontal lobe. The frontal lobe is all about motor control, movement. So remember, Broca's area is the production of speech. So damage to this area is going to lead a person unable to produce speech in a clear or fluid way. Over here, the auditory cortex. When you hear something, that information travels to the ear and goes to the brain in this region. So this is your primary auditory cortex. It's in the temporal lobe. Kind of makes sense, right? The ears are over here next to the temporal lobes. But there's a larger area called the association area that is also associated with hearing. And then we get back to the occipital lobe. The very back of the brain is referred to as the visual cortex, or the primary visual cortex. The rest of the occipital lobe is also involved in vision. So if you ever hit the back of your head and kind of seen stars, you kind of get the idea. But your eyes are way up here, right? Your eyes are way up here. And when you see things, that signal travels into the brain and actually goes back to the occipital lobe. And the occipital lobe is the part of the brain that perceives and receives that information. And the way I remember this one is that my grandmother, when my mother was about 5 years old, my grandmother apparently was out ice skating and fell and hit the back of her head. Within a few weeks, she went completely blind. And at the time, this was the 1950s. They didn't have the technologies and the surgical procedures to do what they do now. But likely what happened is that she broke some blood vessels back here in that fall, and her brain, there was a hemorrhage back here, and that pressure of the blood in the hemorrhage caused this part of the brain to die. Right, there was a lack of blood flow to the occipital areas because she lost brain matter, right, in the visual cortex area. She also lost her vision. So the back of the brain is the area responsible for vision. So let me just recap. Occipital lobe is vision. Parietal lobe is sensations. Frontal lobe is motor. Temporal lobe is auditory. Oh, and here we go. Here's the Wernicke's area. Notice that it sort of straddles over a part of the parietal lobe and down into the temporal lobe. Remember, the Wernicke's area is your ability to understand and process speech. So it makes sense, I think, that Wernicke's area is partly the auditory region, right, the temporal lobe, and sensation region, where you're processing all of those sensations. And this is Wernicke's area. So those are the external landmarks on the brain, the important lobes, some of the other structures. Let's take a look now inside the brain. So we're now going to go in what's called subcortical, right, under the cortex. So we're going to dive down into the brain. And here we're going to be looking and noticing some nuclei. Do you remember what nuclei are? Remember a nucleus? We saw this in the last presentation. A nucleus is a group of cell bodies in the CNS. And remember that groups of cell bodies are going to be gray in color. Okay? So when we talk about nuclei or many nuclei, we're referring to groups of cell bodies in the CNS, and cell bodies are gray in color. So these subcortical nuclei are very important regions of the brain, and I mentioned the limbic system a moment ago. So let's take a look. Hippocampus, right, the hippocampus and the amygdala. These are two regions which are part of that limbic system. They are very, very important in forming long-term memories and in your emotional responses to things. So damage to the hippocampus or the amygdala, these regions would lead someone to having long-term memory problems and having emotional responses that are inappropriate. What's really interesting about this, I think, it's one thing just to learn the parts of the brain, and that seems kind of academic and ho-hum and who cares. But what's really interesting about this is when you begin to understand that if a person has damage to their brain, has a stroke or some sort of trauma to the brain, and certain areas of the brain are affected, the outcome is very different. So for example, if a person has damage to their frontal lobe, they're going to have issues with movement. If they have damage to their temporal lobe, they may have hearing issues. If they have damage to their occipital lobe, they're going to have visual issues. And it gets even more complex as we go into the brain and we see if there was damage to the hippocampus, right, the inability to have memories, and amygdala, the emotional responses would be all screwed up. So these videos go through and describe to you the location of the hippocampus and the amygdala. In addition to those two regions, there are what is referred to as the basal nuclei, right? The basal nuclei. And these basal nuclei are sort of like the checks and balances of your movement. Okay, and within the basal nuclei, there are three parts. They help control movement. Now, notice they're controlling movement, not initiating movement. When you decide to move your muscles, let's say you move your biceps brachii, as you flex your biceps brachii, that information is coming from the motor strip. It's coming from the frontal lobe, that strip called the precentral gyrus. That is initiating the movement. These areas of the basal nuclei are helping to control the movement, making it smoother, making it more fluid, controlling that movement, okay? And these three regions, and I'm not going to have you label these, I'm just going to say them, so I'm not even going to highlight them. The caudate, the putamen, and the globus pallidus. Crazy words, right? I'm not going to have you worry about this right now. I'll show you to where they are. And I just want you to see as we cut into the brain. So let's get our orientation here. We've sliced into the brain. This is basically a coronal section, right, or frontal section of the brain. We can see from the artist that the cortex, right, the gyri on the surface of the brain, we see the darker color, so that's the gray matter. There is that deep longitudinal fissure that separates the left and right hemisphere. Bear with me, I just want to point this out. This band of tissue right here that connects the left and right hemispheres, this is part of the corpus callosum. Inside the brain, we'll see this in a few minutes, but there are large fluid-filled regions. These are ventricles. I did say that word last time, and recall that ventricles are filled with a fluid called Cerebrospinal Fluid Csf. Do you recall from our conversation last time what cells produce this cerebrospinal fluid? One of the glial cells. Recall those are the ependymal or the ependymal cells. So these ventricles would be surrounded or lined with these ependymal cells, which are making the fluid. And this fluid in the ventricles is connected one to another. So this fluid is moving around and through the brain. What I want to point out to you now, though, is, well, let's do one more. This deeper groove right here, this is the lateral sulcus. The lateral sulcus. Remember, the lateral sulcus separates the temporal lobe down here from the parietal lobe and the frontal lobe. When I talk about the basal nuclei, what we're referring to are these three sets of dark matter. Remember, the inside of the brain is mostly white, not completely, but mostly white matter. And that means that it's myelinated axons. Ooh, while we're there, tell me what cells make the myelin that is covering the axons here within the brain. What cells are making the myelin that protects the axons inside the brain? And I hope you said oligodendrocytes. Remember, the oligodendrocytes make the myelin in the CNS, whereas the Schwann cells make the myelin in the PNS. Since we're in the brain, we are in the CNS. So taking a little slice through here, again, I'm not going to have you label this, but you see these darker regions. These darker regions are each a nucleus, right? A nucleus. And we see the caudate nucleus, the globus pallidus is a nucleus, as is the putamen. So all this tells us is that these are clusters of cell bodies inside the brain. And remember what happens at cell bodies? That's where synapses happen. So where you have cell bodies, you have neurons sending in and reporting to those cell bodies within these regions. So again, the basal nuclei, these three regions are helping to coordinate your movement, not initiate, but to coordinate. And I am going to put a big blue section over this conversation because I'm not going to hold you responsible for the direct or the indirect pathways. I'm not going to hold you responsible for this figure as well. So don't worry about any of that. I just want you to know a basic idea that the basal nuclei, right, they are all about, let's find that key word, the basal nuclei are all about controlling movement. Controlling movement. Not initiating, but helping to control it, make it more fluid, make it more regulated. These are regions of the brain that get affected by and in diseases like Parkinson's disease. So if you know someone with Parkinson's disease, right, they don't have enough of the correct neurotransmitters being released by some of these regions of the brain. So when they move their body, they have shaky, right? They have what's called ataxia, or they have shaky movements. Again, the movements are not very fluid. They're not very well relayed or controlled. So think of these basal nuclear structures as regions of the brain that help to smooth out and control the movement. Again, we're going to also cross this section out. So here's a more story, right? The Parkinson's disease and disorder of the basal nuclei, and it gives you a little story about this disease to help you better understand it. So that we've talked about the structures of the external cortex, where we looked at the lobes and the Wernicke's area and the Broca's area. Then we looked deep into the cerebrum and we saw these clusters of cell bodies, these different nuclei, hippocampus, amygdala, and the basal nuclei. Those were all in the Telecephalon, or in the cerebrum, subcortical. Now we're going to go down into that pit, the core, the center of the brain. And this is the Diencephalon. The Diencephalon is deep to the cerebrum, right? It's deep to the cerebrum, and it contains three parts. There is the thalamus, there is the hypothalamus, and there is the epithalamus. Okay, and we'll see that epithalamus is a very small area. It does contain a gland called the pineal gland, and we'll see the pineal gland again when we take a look at the endocrine system. The pineal gland is a gland found in the epithalamus. I'll go ahead and tell you now. You can write it down. But the pineal gland is the gland that makes melatonin. You may have heard of melatonin. It's a hormone, not a hormone, but a hormone that helps to regulate sleep patterns, and it is produced here in the pineal gland. So let's talk about these three regions of the brain. Not hugely at all, but just a little bit of information. The thalamus is oftentimes referred to as the gateway to the cerebrum, or what I want you to see is that it is the relay station. Okay, so the thalamus is going to sort of be the relay station between your cortex and the rest of the body, be it the peripheral nervous system, the spinal cord, or the brainstem. All sensory information, except for the sense of smell, passes up through the thalamus before going up to the cortex. Think of the cortex as the voluntary or the conscious part of your brain. If nervous information gets to the cortex, you are aware of it. You are consciously aware of it. There's a lot of information, however, that never gets to your cortex. It is instead being dealt with and processed by your brain stem. So if things don't go all the way up to your cortex, you're not aware of it. But your brain is working 24/7 to take care of a lot of things. For example, right now, your bladder is sending a signal to your brain. You're not even conscious of it because your bladder is not full. Your brain stem is intercepting that signal from your bladder and saying, okay, everything's good, everything's good, everything's good, everything's good. But when the bladder begins to fill to a point where you need to do something about it, now that signal travels beyond the brainstem, goes up into your cortex, and you now become aware that you need to start looking for a restroom. Right? So the brainstem is controlling or regulating many of your unconscious things, and if it gets up to your consciousness, it's gotten up to your cortex. The hypothalamus is a region just below, right, inferior and slightly in front of, slightly anterior to the thalamus, and we're going to see the hypothalamus again in the endocrine region. It is actually a collection of nuclei that are largely involved with regulating homeostasis, and that homeostasis is maintained by a number of hormones. We'll describe a couple of those as we go through the course. So here, let's get our orientation to this diagram. Again, we're looking at the left side of the brain. This is now, though, a mid-sagittal cut through the brain. Let's talk about first what all of this is, right? All the tan part up here, this is all cerebrum, right? This is all the cerebrum. This is making up the lobes of the brain. As we go deep, this band of white matter right here that I'm pointing to, it's not labeled, but this is the corpus callosum. The corpus callosum, again, is a white band. It connects the left and right hemispheres. You'll see this when you do your brain dissections. Underneath, this is how I think about it, underneath the corpus callosum, this area is the diencephalon, that pit, the core, the center of the brain. Green area is the thalamus. The area below the below and a little bit in front of the thalamus is the hypothalamus. Okay. And hanging down from the hypothalamus is another structure that you're going to be seeing quite a bit of, and that is the pituitary gland. Okay. Now the pituitary gland, you know a little bit about where it sits in, or how it sits, or what structure it sits in the brain. So if you were to open up the top of your cranium, pull your brain up, and look at where the pituitary gland is protected, you would see that the pituitary gland sits in the sella turcica. Remember, the sella turcica is one of the bone markings of the sphenoid, and it's this little indented saddle region, if you will. And the pituitary gland sort of sits down in that sella turcica and is protected by that bony marking. We also have here this oval area. This is the pons, and then this, where I am now, would be the medulla oblongata, and then inferior to all of this would be the spinal cord. Again, you're going to get to memorize or learn all of these parts. Now, here's another video on the limbic system, and it will once again remind you of the structures that are marked here. That is the amygdala and the hippocampus. I'm not going to have you label it from here, but let's just take a look. Here is that corpus callosum. Now it's labeled on this particular diagram. Here, see these indented areas? These are some of the ventricles. So this is a lateral ventricle, and this is part of the third ventricle. You'll see this in another drawing coming up. But these hollow areas, imagine you had this half of a brain laying on its side, and you poured water on top of it. You can imagine that water would collect in this space, right? This is one of the lateral ventricles, and water would collect right around the thalamus, and that is the third ventricle. Here we see the thalamus. Right, here's the thalamus. The blue area is the hypothalamus. And the artist has also labeled the pineal gland back here. Remember, the pineal gland is in this little region called the epithalamus, on the thalamus, right? Epi means on, so the epithalamus. Hanging down from the hypothalamus, here is the pituitary gland. It's not labeled, but there's the pituitary gland. Of interest here, also here's the amygdaloid, or the amygdala, same thing. The amygdala, or the amygdaloid body, right here. And this purple bulge right through here is the hippocampus. The other structures let's not worry about, but those are things that we've talked about so far. So we've gone from the forebrain. Remember, the forebrain is the Telecephalon, the lobes of the brain. You might call it the cerebrum. And the Diencephalon, the pit, the core, the center of the brain. Now we're going to head down to the brainstem. The brainstem collectively is both the midbrain and the hindbrain. The hindbrain is the pons and the medulla. Okay, so all of this together we might just call the brainstem. The brainstem. So again, watch this. Now, when I think of the brainstem, this is the very, very, very important parts of your body, of your nervous system. But everything down here in the brainstem is involuntary. None of this brain matter, brain activity that happens in the brainstem is something over which you have any voluntary control. The regions of the brainstem are the areas controlling your breathing rate, controlling the rate of your heart. This is the part of the brain that is sort of controlling so many other homeostatic functions. A person who has a stroke or damage to the brainstem likely will have issues with breathing, have issues with regulating their heart rate, okay, and their blood pressure, all of that is being controlled here in regions of the brainstem. We'll learn more about those as we go into those individual systems, but just know that that is what's happening here in the brain stem. There's also a number of nerves that come off the brainstem, and if those nerves are damaged, you can also affect problems with those functions. We'll come back to those cranial nerves in another session coming up. So taking a look at this image, right, you should now be able to recognize these things. This would be the cerebrum. Here's the cerebellum. In here would be the midbrain region, which again, I never have you label. It's sort of this little connector area. Here we see the pituitary gland kind of hanging down, just to give us some perspective. Now, sitting right across the street from the cerebellum, this is the pons. And then this other bulged area is the medulla oblongata. Think about it, guys, right here. This is the occipital bone, isn't it, coming around? What opening would be right here? I hope you're all saying the foramen magnum, right? The foramen magnum is that large opening in the occipital bone that allows the spinal cord to pass through. So let's just kind of make a clear understanding that right here, where I'm drawing a line, anything above this line, if it's surrounded by the cranium, this is going to be medulla oblongata. Anything that is poking through the foramen magnum that is now down and protected by the vertebral foramina, we're going to identify this as spinal cord. So let that opening, the foramen magnum, be the border between the brain stem and the medulla oblongata and the spinal cord. In the midbrain, really important things are going on in here, but to be honest, I'm not going to focus on them. So I'm going to do this. I'm going to give you a pass on the midbrain. I'm not going to worry about the tectum, tegmentum, or the inferior superior colliculi, right? Just kind of grab this area, and we're just going to say, you know what, it's important, but let's do this. Just know that this is all part of the brain that is, oh, I can't do more than one paragraph at a time, can I? I always forget that. So I can do this paragraph, call it blue, and I can take this sentence and I can blue that out as well for us. But just know that this is all part of it's connecting, right? It's connecting the brain stem up to the cerebrum. Let's take a look at the pons. The pons is derived from the word for bridge. It's basically sort of the bridge as we think about information coming up through the spinal cord. It also connects over to the cerebellum. Okay, so the pons is the bridge between the cerebellum and the brain stem. That's why it's called the bridge, right? The pons. It is, again, a very important region for basic life-sustaining things. We'll see the pons pop up later as regulating your breathing and things like that. The connectors between the pons and the cerebellum are called the peduncles. That's a fun word. So we'll see the peduncles in the lab when you're labeling some of the lab structures. Then we get down to the medulla oblongata. It is that region that I refer to in the embryological development as the myelencephalon. And myle kind of looks like the word myelin, doesn't it? Okay, so this myelencephalon, it is white, right? Myelin is white, and that's where it gets its name. And this is truly the connector between everything coming up the spinal cord and everything going up into the brain. That makes sense. That's all these axons, right? It's all these myelinated axons that are traveling up and down through the medulla oblongata. Last part to describe is the cerebellum, the little brain. Although it's very small, it is sort of a miniature brain in that it is covered by gyri. right? It's covered by gyri, has little sulci just like the cerebrum, and it's an amazingly dense region. It accounts for about 10% of the mass of the brain, so it's a relatively small region, but there is an amazingly high density of neurons found inside the cerebellum. And the cerebellum is an area, we see it here, right? There it is in this view here. But the cerebellum has a lot to do with movement as well. So walking, I'm just going to highlight walking and balance as two key words for you to remember. Again, the movement of your body is the movement of walking is initiated by your frontal lobe, but the cerebellum helps to coordinate all of that. So a person who has damage to the cerebellum is also going to have balance problems, is going to have issues with walking and other things, because the cerebellum is helping to sort of make that fluid or make it smooth. I'm not going to go into any greater detail than that. I just want you to know that that is the case. That's the main job of the cerebellum. So we've gone through the parts of the brain. Let's now head to the spinal cord. And the spinal cord is going to be, again, continuous with the medulla. It is a part of the central nervous system, and you know that it travels down those vertebral foramina of all the vertebra. Travels down what's referred to as the canal, the vertebral canal. But what you may not initially understand or appreciate, that the spinal cord actually ends where I'm pointing. The spinal cord itself comes down and stops, or the majority of it, what we call the spinal cord proper, stops at about L1. Now, how do I know that this is L1? And the way I easily tell that this is L1, remember that you have 12 thoracic vertebrae, and I can see here rib 12. Right, here's the little floating rib. Here's rib 12. Here's rib 11. So I know this is T11. I know this is T12. The next vertebra down here is L1, right? The first lumbar vertebra. So there are 5 lumbar vertebrae before you get to the sacrum. So the spinal cord ends at or about L1 at a space or a region called the conus medullaris. You'll see that coming in a minute. Coming off the spinal cord are all of these nerves, and we're going to talk about those in another conversation. And we have these nerves coming off the spinal cord. Some of them go down the arm, others just wrap around the chest and the thoracic region, others come off and go around the abdominal region, and then other nerves, of course, come off and head down to the lower limb. We're going to be learning a lot about those spinal nerves in another conversation. If you were to describe the spinal cord, it is not a perfect cylinder. It's almost like you took a piece of Play-Doh, rolled it out between your hands, made like a long snake, right? But this long snake has 2 flat sides, and there are 2 grooves that travel through the spinal cord. Finally, there's actually a hollow center down the center of the spinal cord. So let's look at these words, and then we'll look at an image where these are labeled. So there is a line on the front side or the anterior side of the spinal cord, easy, called the anterior median fissure. What does that tell us? Right? That it's in the front, anterior, it's down the middle, median, and it's a groove, a fissure. Don't make this hard. On the back side, there's a posterior median sulcus. Okay, what did I tell you the difference was between a fissure and a sulcus? Remember, a fissure is a deeper groove, and you'll see that to be the case, that on the anterior side, the groove is deeper, so it's a fissure. On the posterior side, the groove is not as deep, it's more shallow, it's a sulcus. Coming off from the spinal cord, there are axons that travel off. Some of them travel off the front side. Those are the ventral or the anterior nerve roots. And there are others that travel off the back side. they are the dorsal or posterior nerve roots. This will make more sense in a picture here in a moment. These nerves that are traveling off the spinal cord carry both sensory functions and motor functions. So we'll see that these spinal nerves carry both sensory and motor function. And here are a couple more words, and then we'll look at a picture, okay? So the spinal cord comes down, and it ends at approximately L1 at a tapered end called the conus medullaris. I want you to imagine a pencil, a sharpened pencil, and you're holding the pencil by the eraser, and the pencil is hanging down, and at the bottom you have the point, right, where you write with. So the spinal cord is the length of that pencil. Where it tapers off at the point, that is the conus medullaris. But even at L1, at the conus medullaris, there are additional nerves that are going to come off the tip, and all of those nerves are called the cauda equina. Cauda, you recognize as a term meaning tail. Equine, horse. It kind of looks like a ponytail or a horse's tail, hence its name. Let's do a couple more images or a couple more words, and then we'll put all this together in a labeled image. As you look at a cross section of the spinal cord, you will see what I describe as a butterfly. Others here describing it as the capital letter H. Okay? And when we look at this letter H, or what I'll call the butterfly, we're going to see some gray matter. And this gray matter is broken up into regions called horns. So the gray matter of the spinal cord is broken up into areas called horns. That's just a different word, right? Just horns. Weird word. Okay. We're going to see that the posterior horn is all about sensory. The anterior horn is all about motor control of your skeletal muscles, and the lateral horn goes out to your autonomic nervous system. Remember, the autonomic nervous system are those things which are happening for you automatically. Okay, so let's take a look. Here is a cross section through a spinal cord. This is an artistic rendition. Here's an actual slice through the spinal cord. Now it's been stained with some purple and pink stains, but regardless, you can see this letter H, or what I call a butterfly. As we look at this butterfly with the wings of the butterfly, right, are broken up into areas called horns, right? So we have a horny butterfly here. We've got the posterior or the dorsal horns. We have this little area sticking out of the side. This is the lateral horn. And we have here the anterior, also called the ventral horn. Okay. In the very center is a strip of land here. It's not labeled on this image, but it's coming up, called the gray commissure. So the gray commissure is connecting the two butterfly wings together, and in the very center of the gray commissure is a hole called the central canal. It's traveling up and down the entire length of the spinal cord and is filled, like ventricles, with cerebrospinal fluid. Notice here, let's get our orientation. Whenever you see the gray butterfly, the wings always reach up and touch the back side. So the way I get my orientation, this is definitely the posterior side. See how the wings are coming up and touching the very edge. Notice on the anterior side, you don't have the horns touching. There's a gap, okay? So whenever you see the wings, which ones are longer, reaching to the back, let's look at this one. You can see that the posterior horns are reaching up and touching the actual edge of the spinal cord, whereas on the anterior side, they are not doing so. And this is the groove. This is that anterior median sulcus, not as deep as the posterior median fissure. So you can see this is a deeper groove. It's not so obvious here, but it's a deeper groove. You can see it here. It's a much deeper groove on the posterior side, hence the name fissure. In addition to the gray butterfly and these regions called horns, there are also the regions around it. Now this is white matter, right? So everything on the sides and on the front and around it is white matter. Think about it, guys. This is backwards from what we saw in the brain. In the brain, gray matter was in the cortex, right? It was on the outside edge. White matter was on the inside. So what we see is that the arrangement of white matter and gray matter is flipped between the brain and the spinal cord. In the spinal cord, the gray matter is the butterfly. The white matter is surrounding the butterfly. These regions of white matter are called columns. Columns. So here we have the posterior columns, on the side the lateral columns, and in the front the anterior or ventral columns. So again, this is not hard, it's just new words. The white matter is called columns, the gray matter is called horns. Now, there's another name for the columns. So let me find the best place to add this note. The columns are also referred to as, let me just do it here. The columns are also referred to as funiculi. Each one is a funiculus or many funiculi, okay? And so this is just another name for the columns, and you'll see this word in various places, including, I believe, the Green Lab Manual. So what is going on? In the white columns, it's white because it is primarily axons, right? Traveling up and down the spinal cord. These axons are in groups, and you remember what a group of axons is called? A group of axons in the CNS, remember, is called a tract. A tract. So remember, a tract is a bundle of axons in the CNS. What is the equivalent term? In the PNS, right, we would call this a nerve. But in the CNS, we're going to call it a tract. So we have bundles of these axons traveling up and down through the spinal cord. Ascending tracts are going to be sending sensory information up to the brain. Think about it, right? The ascending signals going up the spinal cord. They are sending sensory information from the periphery up to the brain, whereas the descending tracts are sending motor information down from the brain that are going to tell the body to do something, right? Tell a muscle to move or tell a gland to secrete something. And then there's, and we see that these areas, right, the posterior columns and the anterior columns are dividing up these different tracts. I'm not going to have you label those. Just know that the columns, right, the white matter, are carrying ascending and descending tracts of information. And that brings us to the end of this. Now, I know you're probably feeling rather unsatisfied. We're going to continue to further understand what these different regions of the brain are doing. For now, however, your primary job is to learn to label the parts of the brain and of the spinal cord. Okay, that's your takeaway. That's going to be your lab-focused thing you're going to be doing. I gave you a little bit more information here, and we'll continue to pull this all together.